So why don't we go ahead and get started? Uh, Welcome to Science Matters. My name's Amy Dudley, and I am a PI and the Chief Scientific Officer here at the Pacific Northwest Research Institute. And I'm here to welcome you to a special edition of our Science Matters seminar series. September is National Newborn Screening Awareness Month. And in recognition of this important public health program, each year we host a special format of Science Matters in which we pair scientists whose research focuses on some aspects of advancing newborn screening with other members of the rare disease community. And today we have a wonderful pair of speakers for you. We're going to switch up the order a little bit because we have one of our special guests is Dean Suhr, who's going to present some of the impact and some of the kind of justification for why some of this work is so important. So Dean is the president and co-founder of the MLD Foundation. His career began in high tech, where he spent almost 2 decades as an executive in engineering, operations, and business development. And then when 2 of his 3 daughters were diagnosed with MLD in 1995, he and his wife co-founded the MLD Foundation, which for more than 25 years has supported families and researchers worldwide. Dean's played a key role in advancing therapies, including a curative gene therapy that just received FDA approval last year. And he's also been deeply involved in newborn screening efforts. He's collaborated with our second guest, Michael Gelb, since 2008. And more recently, they've launched the MLD Newborn Screening Action Alliance to shape the future of rare disease screening for the next generation. So Dean, welcome.
Thank you. And we'll save the smart guy till last. But I hope that what I share today will be valuable for all of you. We're going to have some Q&A. We've reserved time at the end. If there's a pressing question, midstream or maybe after my talk, we're happy to jump into that. But I do want to make sure that we reserve time for Mike and his presentation as well. So let me get us started here. Look at that magic of high tech when it works. I love that. So it's always interesting when you title a presentation and you're a month or 6 weeks ahead whenever we started organizing this, and then you come back to it and you say, well, what was I thinking and how was I thinking and so on. And I actually didn't change much from what my initial thoughts were. So I hope this is informative to you. I am, as Amy said, I'm an electrical engineer, so I understand technology, but I've been involved a lot in business development and marketing and sales and reliability and all kinds of those sorts of things. So across a lot of departments, and I think you'll see that as I go through the presentation. But most important, I'm a rare disease dad. As Amy said, 2 of my 3 girls have MLD. Darcy, who was 10 in 1995, she had an experimental bone marrow transplant really, really early in using bone marrow transplants for MLD. It was just coming over from the leukemias in the oncology space. Lindy, who at the time was 14, we had a 6-year diagnostic odyssey with her, which we're going to talk about newborn screening today. Hooray, people won't have that long of a diagnostic odyssey, which will be wonderful. They told us Lindy would live another 4 to 6 years into her early 20s. She turns 45 this weekend. And while she's full care, she's doing much better than the papers. We didn't really have an internet back then, than the papers and publications said. So this is part of my motivation, but my involvement really is much, much broader in the newborn screening and the rare disease spaces. So I want to just kind of lay some groundwork for you. We at MLD have what we call the trifecta. We have an approved therapy. It's curative when the diagnostics, in this case newborn screening, are used to identify those patients pre-clinically symptomatic. Notice I said clinically symptomatic because this disease starts at birth and probably in the womb. And then the third piece of that stool, so that you have all the pieces you need to take care of these babies, is you need access and reimbursement. And that's physical access, geographic access, as well as, in our case, gene therapies, as you've probably read or are aware of, are pretty expensive. And so you need to have the reimbursement. And I'm going to touch on that very briefly. Very quickly on our timeline, and I'm not going to spend a lot of time on this, but we formed the foundation in 2001, again, diagnosed in 95. and were primarily family focused until about 2005. We met with some what other people called crazy gene therapy researchers. 2007 started our federal policy work. 2008 we started working with Dr. Gelb on the MLD assay. Remember that 2008, 17 years ago. 2013 clinical trials started in Italy. This therapy was developed in Italy. Gene therapy was approved by the EMA, the European equivalent of the FDA, in 2020. So from the start of clinical trials till then was 7 years. So that was a long journey. First patient received gene therapy in the US in Minnesota in 2021 as a compassionate access patient. We didn't have the therapy approved yet. We received very positive value assessments, independent assessments by ICER and NICE. ICER is a US organization. NICE is the UK National Health System, and they looked at what they anticipated the price might be of this therapy and how effective the therapy was. Again, when these patients were caught pre-symptomatic, and very, very positive indications from them. 2024, last year, was a big year for us. We got the gene therapy approved by the FDA. It took a little bit longer because the data was collected in Europe and they do things a little bit differently over there. And because of our history, there was some data massaging. There was no additional studies that were needed for that. And we submitted our RUSP nomination. And if you don't know what the RUSP is, we'll get into that in just a moment. But that's the start of being formally recognized as a newborn screen that should be implemented by all of the states. 2025, this year, last Friday on 9/12, New York became the first state to do population-wide newborn screening for MLD. The first of many, we hope. And as of this point in time, we have 5 gene therapy QTCs, qualified treatment centers. So this gene therapy is not available at your local drugstore. You've got to go to some specialty centers for that. And they're spread out across the nation. So where are we as we sit here in late 2025? We hope MLD will be added to the RUSP. That's not done yet. That's why the question marks are there. But as we'll talk about in a few minutes, the RUSP, the Recommended Uniform Screening Panel, the federal recommendation, not requirement, the clock starts on 53% of the babies that are born in the US. The states that serve those populations are required to start looking into screening for MLD or anything else added to the RUSP. In addition, in the MLD community, we have 10% additional babies in process at 4 more states. But it will take years to get all of these programs up and running because, we're going to talk about this in a moment, newborn screening is public health and it's state public health. So it's a 60-year-old program, started with PKU back in the 60s, and has been very, very successful. 98% of the nearly 4 million infants born each year in the US are screened. It is an opt-out program. So most people, any of you that have kids or have family members that have kids, probably don't even remember your newborn screen. It's a heel prick. It's a drop of 5 drops of blood that come out of the baby's heel. And if nothing is wrong, you never hear about it. That's a good thing and a bad thing. It means that when something does go wrong, people said, well, I didn't know you were testing for that, and on and on. But that's another topic. About 7,000 infants a year are identified for intervention. And the network, I said, is the 50 states, but it's actually 56 states and territories. And there's actually, some of the Indian and Native American nations and so on that have their own independent public health systems as well. So there's a lot of moving pieces. It is the nation's most successful, most equitable, and cost-effective health program. It's very cost efficient to screen everybody at birth. The systems, they're used to screening millions and millions of blood spots. And it's most equitable because 98% plus of babies see this program. There aren't fiscal concerns. There aren't, what district you live in or racial or religious or any of those sorts of issues. Newborn screening uses federal support, guidance, and appropriation. So there's a federal role in newborn screening as well as what the states are doing. The state autonomy allows the states to optimize for their state's needs. We've been doing some work with folks up in Alaska. Their needs are distinctly different than Pick a small state, New Jersey, where you can drive from one end of the state and the other in an hour or two. That doesn't happen in Alaska, so the mechanisms are different, and so the states can respond to that. But it also leads to variances in the resources and practices. Who has the funding? Who has the expertise? Some states screen a lot of samples. Some states screen a significantly smaller number, and so the expertise and capabilities vary across states. And one of my advocacy peers calls this death by zip code. So if you're in one of those premier states for whatever reason, then everything goes generally everything goes really, really well. But if you're in a smaller state or the state hasn't started screening for a particular condition, you're over a line on the highway or a bridge or a river or something, and that difference could mean whether your baby is detected for a disorder. Newborn screening only screens for disorders that have therapies, and you must have, of course, an efficient and accurate screen. But you also, the disorder has to be something that wouldn't otherwise be detected by the pediatrician through some of their normal new baby visits and those sorts of things. So there are criteria for how you get to be a newborn screen. Where we sit today, the technology, the therapy, the ethics, frankly, the politics need to prepare and adapt. And I say prepare, I just mean that we need to really adapt to not only what's happened, but what's happening and where we think we will be in 5 or 10 years. Newborn screening ecosystem. So many so many people think this is the ecosystem, the baby in the lab that does the testing, and that is just not the case. My friend, the director of the lab in Texas, Susan Tanksley, she created this diagram many years ago, and I just love it. If you look at it, I don't know who's not on this list. Everybody is involved. You see the advisory committee, the ACHDNC in the lower right. You see policymakers and parents in the upper right, the insurance companies, the hospitals. And a particular group I'm particularly involved in, which I think is lumped into stakeholder groups, is the advocacy groups. And we kind of blend across all of these things. But everybody in this ecosystem has to be informed, has to be working in lockstep, and has to participate in implementing a newborn screen, a new newborn screen, and in supporting the system as it works day by day. But 2025, as we all know, on a whole bunch of different levels, is an interesting year. We've had big shifts and changes, specifically at HHS. Health and Human Services is one of the departments, like the military, Department of Defense, Department of Education, and so on. Health and Human Services is the federal government's department that is responsible for the NIH, the FDA, the CDC, and HRSA. You've probably heard of the first three. The next one, Health Resources and Services Administration, is part of HHS, and that's where newborn screening resided. And today it resides, but we understand things are moving around inside of HHS, so we'll see how that all works out. But all of our history is with HRSA. All of these changes create uncertainty, confusion, and opportunity. And I know many of you are in the research space, and so you're looking at funding and programs and all those sorts of things. And certainly uncertainty and confusion, I get it. But it also creates opportunity. And I encourage you, no matter where you are, to be thinking about how can you pivot and adapt and seize the moment, so to speak, because changing the direction of government is not something that one person can do. We should all be impacting that as we can. But you in your lab, think about what are the opportunities that are coming from these changes. Specific to newborn screening, the ACHDNC is the Federal Advisory Committee to Health and Human Services, to HRSA and Health and Human Services, and it's called the Advisory Committee for Heritable Disorders in Newborns and Children. So that's why we call it ACHDNC. It was disbanded. It was a provisional committee because some of our legislation didn't get renewed about 5 years ago, and we've had trouble renewing it. So instead of being a permanent committee, it was provisional, and it got swept out, not because it was newborn screening, but because it was a provisional committee. The RUSP, the Recommended Newborn Screening Panel, Recommended Uniform Screening Panel, sorry, I've extended that out improperly. That is the key document that is a federal recommendation to states and territory health systems to implement newborn screening. They do kind of a one and done expert review. They have criteria. They understand how challenging some disorders are in terms of implementation, in terms of making referrals and follow-up and so on. And they make a recommendation if they think that all of those problems have been addressed. They make a recommendation to the Health and Human Services Secretary to formally get added to the RUSP. The RUSP still exists. It didn't go away, but it's basically frozen because that committee's not there and it can't add new conditions. As I mentioned, I think on one of my earlier slides, RUSP alignment, the RUSP covers about 53% of newborns born in the U.S. RUSP alignment means that states are obligated by state law. We don't include those that do it by practice, but by state law to when a disorder is added to the RUSP, to this recommended panel, that they consider the disorder. A couple of states have to implement it. Most of them have to consider it. So the challenge with us right now, there's no organized way to add new disorders and to guide the ecosystem, to be efficient. And again, I use the word efficient in the federal government in the same breath here, but to be efficient about what the federal government can do once and well, and then don't have to repeat that at the state level. Sequencing, Dr. Gelb is going to be talking about sequencing and some of the science of this, but it's making bigger and bigger impacts, not only in the screening, but also in the referrals and how babies are directed to the appropriate therapies at the appropriate time. Blood spot retention is and has been a privacy concern. The government's got my DNA or got my baby's DNA, and sequencing only adds to this. This has been part of our legislative challenges. Really has nothing to do with newborn screening. It's this perception that that the government having a blood spot or sequencing data is the end of the world. We need to be honoring privacy. I got that. I understand that, but we also need to have good public health systems working too. But some of what went away can stay away as far as I'm concerned. And I'm an advocate and I'm not, I'm not loyal to anybody, I guess. I say what's right for the families, what's right for the system. There were things about about that advisory committee, and there are things about newborn screening that we think we can do better. And disruption is a time to pivot and take advantage of that. And this is my little plug. All of you should be aware of what's going on with policy. And those letters or those phone calls, those quick emails, 1 line, 2 line, 3 lines, where you express your voice on anything relative to policy is impactful. I do this probably 25% of my time, and I know it's impactful. There was a report that came out earlier this year from the National Academies of Science and Medicine, a newborn screening report. And while Dr. Gelb is talking, I will post this link into the chat. I can't talk and look and paste at the same time. But this independent report was put together after a year-long study of experts and KOLs inside the newborn screening ecosystem. And there are a number of things that are in that report. Some of this is beating the same old drum, but at least it's formally in a report, and there was general consensus around this. They're asking for a number of structural things, but notably on the bottom left, prepare and adapt for the future. We need to be looking forward to how is technology and how are therapies and how we, I say the patients and families and the researchers and doctors and clinicians, how are we communicating and sharing information? We need to be thinking about that with regard to newborn screening. And then a whole bunch of additional national coordination, which generally I'm in favor of because it's more efficient. So I talked about pivoting. The ecosystem and international collaboration, that's entirely different than it was 10 years ago. We've got legislative mandates where advocacy groups, and I admit that we've done a little bit of this in MLD. Sometimes it's helpful. Sometimes it's walking alongside the government. Sometimes it's doing a little bit of prodding, and sometimes it's leading. It just depends on the state. And then in the MLD, metachromatic leukodystrophy, by the way, is a leukodystrophy, which my 2 kids had. It's a neurometabolic disorder. It is genetic in nature, and it's terminal if not treated. And there are infantile forms all the way on up through juveniles and into adults. The DMD is Duchenne muscular dystrophy, a little more complicated disorder. I am not an expert in DMD, but both of us were going through this process with the advisory committee of their expert review when the committee went away last, I believe it was early April. And both communities were supposed to be voted on in May to be put on the RUSP, and that rug was yanked out from underneath us. Those reviews, those expert reviews were completed. Ours was all but spotless. And we are now working with the Secretary Kennedy at Health and Human Services to try and get directly added to the rest. Not shortcutting, but just completing the process we started 9+ months ago. So if need be, we in the advocacy community, and this is not just about newborn screening, it's about everything that you do in the research space, but we'll be creative, and if need be, we'll be disruptive, in our case, to save a baby a week in the US. So we're doing that. We're not trying to upset apple carts, and we're excited about the therapy. I should say, just before I finish that, our therapy not only got good value assessment, but the reason it got that is because it's essentially curative when you identify these babies pre-symptomatically. So we are pivoting not only in the MLD community, but in the newborn screening community beyond the NASEM report. I think that's a good foundation and an anchor and a common point of collaboration. But we are working to create a refocused and more efficient advisory committee and screen approval process. The things we didn't like, remember I mentioned on a bullet a couple of slides ago, we want to improve those, and we can do that more efficiently with technology and with approaches that we have today we didn't have a decade or 2 decades ago as newborn screening as we know it was being launched. Pilots, new screens, and overall operational data sharing, short and long-term referral and follow-up reporting so we know how the babies are doing. Sequencing, again, I'll leave this to Dr. Gelb. It's a very valuable tool, but it is not black-white. It's complicated, and it's much more than the ACs, Gs, and Ts that you all know a lot more about than I do. I have formed with a partner in Washington, D.C., the Newborn Screening Action Alliance, and the emphasis there being on action, with some differentiation being action as well as appropriations and funding, because that's one of the things that's not being well addressed. So that's one of those pivots and seize the moment opportunities. And again, this is not a pitch for that, but just to give you an idea that people are doing things to try and make this better. And with that, again, I think unless there's burning questions, I can't see the chat, but if there's burning questions, pipe those up. If not, let's get Dr. Gelb and learn a bit about sequencing, and then we'll come back for a healthy discussion.
Great. Thank you, Dean. That was a wonderful introduction and discussion of newborn screening and your journey through MLD. So our next speaker is Dr. Michael Gelb. Dr. Gelb is a chemist and a biochemist who has developed most of the biochemical screening assays that have been added to the newborn screening panel in the past 10 years. Michael earned his bachelor's degree at UC Davis and his graduate degree at Yale, where he also did a postdoc before doing a second postdoc at Brandeis. He is currently the Harry and Catherine J.N. Bonand Endowed Professor of Chemistry at the University of Washington here in Seattle. Michael has received many awards and honors, including most recently the 2022 Guthrie Award from the International Society of Neonatal Screening. And on a personal level, one of the things that I admire most about Michael is that he's an innovator and a problem solver. He sees bottlenecks and he fixes them. And one example of that is he saw the need for high quality chemical reagents to support newborn screening. So he founded a company called Gelb Chem that's now Enfanos. And more recently, he saw the potential for sequencing-based newborn screening to advance a number of treatable diseases that could be diagnosed early. And so that's when he ventured into the field of mutational scanning, which is part of what he'll talk to us about today. Michael, thanks for taking the time to speak to us.
Okay. Yeah, I should explain. So my students wanted to start a company and they called it Gelb Chem, which I thought was a little goofy. And I ran with it for a while. And then a month later, I said, oh, wait a minute, we got to change the name of the company. So now it's called Enfanos. Anyways, that's my disclosure. Half the people in the world think I named a company after myself. I mean, people that know me would probably think that's possible, but I really didn't. On the other hand, it was called that for a while. So anyways, we changed it now, so I feel better. Okay, good. Anyways, I'm going to play this movie. Can you see it?
Children with late infantile
Okay, so this movie is kind of eye-opener.
onset metachromatic leukodystrophy lose the ability to walk, talk, swallow, eat, and see. Most kids with MLD won't survive beyond their 5th birthdays. In 2010, using stem cells derived from their patient's blood, researchers pioneered a treatment to repair the defective gene that causes MLD. Giovanni Price was the 2nd child in the world to undergo this treatment. Today, Giovanni is 5 years old and has no symptoms of MLD. On August 24, 2015, Giovanni Price did something no child with this disease has ever done before. He walked to school for his first day of kindergarten.
Okay, I thought that's a nice way to start a seminar. Okay, so I'm going to talk a little bit about the technology of newborn screening, sort of a quick summary of the last 10 years, and then some unpublished stuff that I think is kind of exciting and powerful. But let's talk about MLD, metachromatic leukodystrophy. So, there's lots of leukodystrophies, the loss of white matter. I think the reason is because most of your white matter is developed after you're born, and so if you have hemostasis problems in your white matter, it's gonna not be a miscarriage, but an early onset disease. That's typical for diseases versus miscarriages, things that are fatal to the fetus. So there's a lot of sulfatide in white matter, myelin, and it's a sulfated lipid. I don't know exactly what sulfatide is doing. It's part of the membrane, but it has to be broken down in the lysosome. The lysosome is in charge of turnover, breaking things down for turnover of things that are on the surface of cells, not the inside of cells, but the outside. So sphingolipids tend to be on the outer surface of the plasma membrane, and the membrane is always invaginating to form endosomes in the cell, which fuse to the lysosome. So what was on the outside of the cell becomes on the inside of the lysosome, where the metabolites can be acted upon by hydrolytic enzymes, such as the sulfatase that removes the sulfate. And then the ceramide, the galactose is removed and the ceramide is broken down. It's about 1 in 70,000. So I think this gene therapy that was developed in Milan is one of the most remarkable medical breakthroughs in my lifetime. I didn't develop it. I just think it's remarkable. So the thing about lysosomal enzymes is, they go to the lysosome, but they're also secreted from cells. They go through the classical secretory pathway in addition to going to lysosomes. They go through the ER and the Golgi and everything. They get out of cells. You'll find lysosomal enzymes in the plasma, but they can also go into cells by endocytosis, right? The endosome that forms after the plasma membrane invaginates, it fuses with the lysosome. So it was shown, 30 years ago that if you take cells that have a lysosomal enzyme and co-culture them with cells that don't, you can transfer the enzyme from one cell to the other through the extracellular media. So that means we can do things like enzyme replacement therapy. We can inject literally the recombinant you have a broken enzyme. We can give you good enzyme in your blood. And for all cells in contact with the blood, the enzyme can actually go into the lysosome of those cells, right? It never has to cross the membrane. Officially, it's all done by vesicle budding and fusion. But of course, the enzyme doesn't cross the blood-brain barrier, although now we have new enzymes that are fused to things like transferrin, for which you have a receptor that carries it across the capillary wall in the brain. And we can get these enzymes into the brain. But the other way we can get them into the brain is through bone marrow transplantation, because you have a number of cells in your brain that are hemopoietic. They were born in the bone marrow. That you have resident macrophages, for example, in the brain. So we can wipe out your bone marrow. You can do a leukemia thing. We can wipe out bone marrow and then give you bone marrow stem cells from a healthy donor that has good enzyme, reconstitute the bone marrow. And some of these cells will go into the brain and they will spew out lysosomal enzymes into the interstitial fluid of the brain and be taken up by cells in the brain that don't come from the bone marrow. So this is called cross correction. And this works pretty well in some diseases, but for MLD, it doesn't work so well unless you overexpress the enzyme that's called ARSA, the sulfatase, in the hematopoietic cell. So this is kind of a gene therapy stem cell transplant combo. Take cord blood from the baby, isolate the stem cells that are in the peripheral blood, and transfect them with a lentivirus, an HIV virus that's engineered, and it produces large amounts of the ARSA enzyme under a strong promoter. And it's integrated into the chromosome, so it's a permanent thing. It reconstitutes the bone marrow. These cells then find their way into the blood, into the brain, where they can cross-correct other cells in the brain. And by overexpressing the ARSA with a strong promoter, it just works a lot better than if you don't do that. That's where the gene therapy comes in. So this is, I would say, low-hanging fruit gene therapy, because we can't get the gene to all the cells in the body, but we can get it to cells that kind of matter. We can transplant those cells, right? Now, if you're missing a lysosomal enzyme, you might just think, why don't we just measure the activity of the enzyme? And we do that in a lot of cases. We take the dried blood spot, we add buffer, we measure the activity of the enzyme by adding a substrate and let it incubate. And we use mass spectrometry to measure the enzymatic product. And the reason we do that is because we can do many products at the same time because of mass spec, right? You can't do that by fluorescence, for example. ARSA MLD is a little bit different because the enzyme is a little bit unstable, and so we don't measure it in the first round. We do measure the equivalent, which is the accumulation of the sulfatide substrate. So when the enzyme is defective, the substrate accumulates. Now, how do we know it accumulates? Well, we know that in the clinic for many years, but the question is what does it look like at birth? This is where it's hard. So this is where we worked with Dean and the MLD Foundation. So we contact all the families, which we need them to do. They have the registry. And we go to places like California, where they have this very vital resource where they store the newborn blood spots for decades, the leftover. And states are talking about destroying these facilities. That would be the death of newborn screening, because we need to go to the patients that are 5 years old that have MLD symptomatic and go back and get their blood spot at birth. And the only way we can do that is places like California, where they store them. We can get the blood spot and take a look at the sulfatide levels at birth. So we did that, and they're elevated, but not too much. You can see some overlap with the non-MLDs. But anyways, we proceeded. This is Giovanni that you saw in the movie. He's going to high school now. This was a few months ago. He's perfectly healthy, 6'3". This is his mom, Amy, testifying to Congress. Okay, so we have this overlap. So what we do is anybody that has high sulfatides, we then measure the enzymatic activity in the same blood spots. Now, I said the activity is not very stable, but it's stable enough because if you have MLD, the activity is really kind of 0%. We're looking for the guys that are really low. But we do that on top of the sulfatides being high. It just works better that way. And then we sequence the DNA as a third tier to see if we find the mutations. And I'll come back to why DNA sequencing versus biochemistry a little bit, but we do this by biochemical. Now, do we have any false negatives? Do we miss anybody? Well, how would you know? Okay, you could start newborn screening and you could say, well, did anybody come forward in 5 years into the Seattle Children's Hospital that has MLD and we missed it? Well, that's not very reliable. A better way to do it is we ended up getting about 40 newborn blood spots from kids that went on to develop MLD, and we showed that we catch all 40 with our tests. So you'd have to be a cynic to think that we would miss a bunch of babies if we're catching 40 out of 40. What about false positives? Do we find elevated sulfatide and low ARSA in kids that have MLD? And the answer is so far no, out of 500,000 tested. So this is probably the best performing newborn screening test ever developed. We have, as far as we know, no false negatives, and as far as we know, no false positives. False positives are bad. You tell the family your kid might have MLD. It's a horrible disease. Not so good. talk a little bit about, I think let me see before I do this. I want to switch to another deck quickly. No, hang on one sec, stand by. Okay. Now we go to share. Can you see this 10-plex?
Yeah, that's great. If you
Okay.
can put it back into presentation mode, that
So
that
to
would
make a
be
long
great.
story short, mass spec is really nice for multiplexing. We can measure biomarkers like sulfatides. We can measure enzymatic products all in the same 2-minute mass spec run. They all have different masses, and mass spec is good for measuring many analytes all together. So this is a new 10-plex that's been launching in newborn screening labs where we measure a bunch of enzymatic activities directly or biomarkers, like I showed you. The enzymatic product is the same thing. We take the blood spot. This time we incubate it, let the product form, and we just detect that by mass spec. Or we measure the analyte, the sulfatide that was there at the beginning because the enzyme was defective. Okay, so we can do all these at once, and that's the punchline. Can we do 100 diseases? Probably. Can we do all the 600 diseases that they're talking about doing by DNA sequencing? I don't think so. No way. So that's an advantage of sequencing, is we can do things that we cannot do biochemically. But with sequencing comes lots of challenges. Get into that a little bit. I don't think I want to. Oh, maybe I'll show you, the concept that okay, I think that's all I want to say about that. Let me now get into the new stuff. Give me 1 second. Sorry, I'm switching decks. Okay. 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 I want to talk about voodoo. Yeah, we do voodoo in my lab. It means we do lots of VOOs. VOOs are variants of unknown significance. This is what Amy Dudley works on. We have all these missense mutations, let's say, causes amino acid changes. Are they clinically significant? And if we don't have case reports, we don't really know. All we have are biochemical screens, like what Amy does. Very powerful. Show you one version of this. So I'm sort of switching gears. Let's say we screen for MLD and you test positive biochemically. We're certain pretty much that you're going to get MLD. The question is when? Because you see these diseases always come in a grayscale. You have infantile forms, you have juvenile forms, you have adult forms. The gene therapy is approved only for the early onset forms, let's say the infantile and the early juvenile, but not for the adult. Maybe it's coming for the adult. I mean, it's no picnic if your teenager develops MLD, it's pretty bad, but the priority right now are the early onset kids. So the question is at birth, how do we determine prognosis for when you're going to get the disease? And this is really hard. So now genotype's important because there are certain variants that we know are really bad guys. Stop codons, particular missense mutations, frame shifts that, annihilate the enzyme. And if you have 2 of these in trans for a recessive disease, you're going to get an early onset form. But what about all the missense variants for which we don't have data in the clinic? So what Amy can do, and also in my lab in a different way, is we can look at all possible missense variants. All possible means changing every amino acid to all 19 other options. at 1 change per protein. So if you have 500 amino acids, maybe 10,000 missense variants. And, you know, for $10,000 now, a biotech company can make you a library of all possible cDNAs, where every possible missense variant is in the library, so-called saturation library. We take the DNA, put them into a transfer plasmid, and we also add a 16 nucleotide barcode, which there's 4 billion of them, and it's in the same plasmid. So if you sequence the barcode later, you have the hash table, right? You have the phone book. You tell me the barcode, I tell you the missense variant, because they're linked in the same plasmid. And we use PacBio sequencing for the long reads. So we have the phone book. We read all the plasmids, millions of them, and we have the barcode sequence, missense variant sequence linked together. We have that list. It's kind of like Survivor's Island. We send a bunch of people to an island and we have a manifest of everybody on the boat. Now we're going to let them duke it out on the island, and some people are not going to make it home, and we take a manifest of the boat going home and we see who's missing. That's kind of the strategy. It's not quite black and white, because some people go home missing or some people go home with broken legs. So we can tell the degree of misfunction of the survivor. The way we do this is really clever. It was developed by Doug Fowler in Genome Sciences at UW. We transfect special 293 cells. They have a landing pad, which is a unique DNA element at a single site in the genome. There's a bacterial integrase that stuffs the entire transfer plasmid into the chromosome at a single site. Once the first one goes in, it's game over for that cell. So every mutant has a home inside of a cell, because we have millions of cells. But any given cell expresses only one variant. We know that. Okay. Now, in this case, there's 2 ways we can do it. In this case, the ARSA protein is fused to the green fluorescent protein at the C-terminus. If the protein is degraded, we will lose the fluorescent signal, because the green fluorescent protein gets degraded along the way. I'll show you that in a minute. So if we have a way to measure the fluorescence, the green fluorescence in each cell individually, we can find the cells that express mutants that cause the ARSA to be misfolded and degraded. So here's, on the left is the wild type ARSA. It goes into lysosomes with the GFP. You see the punctate staining for the lysosomes. We also express a red protein throughout the cytosol of cells from another protein, and that doesn't go to lysosomes. And we simply measure the green to red fluorescent ratio in single cells by FACS cell sorting. Right? This is a wild type that has a distribution of green-red as a function of cell number. And this is a mutant that causes mostly disfolding. Most of the cells have low green-red. It's not black and white. It looks like this. It's distributions. But let's say we collect the cells that sort at this green-red. These are going to contain mostly cells that have misfolding variants, compared to collecting here cells that are mostly folded. So we can do this quantitatively. I won't go through the details. Oops, why is that there? Okay, so now we take all these cells at any given red-green that we want to, bin. We're interested in the low green-red, and we then just amplify all the barcodes and sequence them by next-generation sequencing, which gives us literally the counts of all the barcodes. So we say, okay, here's 47 reads of the wild type, barcode, but this barcode we only see once. So this mutant, alanine, 129, serine, whatever, doesn't fold very well. And it's quantitative. It's present 1/47th as much as wild type. It's that simple. So we can normalize to wild type. We can tell you the survival frequencies for all the variants. Okay. What I was going to show you, maybe it's in the other slide deck, which I won't. So one thing you can do is we have all the variants. So you might turn this loose on all the variants in ClinVar that are classified as, let's say, pathogenic. And by and large, it gets this right about 98% of the time. There are exceptions, right? This is not perfect. So what if you have a variant that messes up the active site residue but doesn't cause misfolding? That would be clinically significant, but we would read it as surviving. Okay. So we're left with sort of the game that, let's say your sulfatides are high and your ARSA is low. And we do the genetics and we find, let's say, one bad guy and one variant of unknown significance. It's not in ClinVar. So if you have a very low folding score in our assay, we're going to worry a lot about this patient, even to the point that they would have an early onset disease. We're going to follow them very carefully in preparation for gene therapy. If the folding score is high, we cannot exclude MLD because it could be a rare case where you mutated the active site and it still folds. So there's a caveat. But most of the time it's a folding problem, so most of the time this works. Now, what if you're in this category of having high sulfatides and your folding score is okay? We're still going to worry about you. We're going to do other things like enzyme testing and leukocytes. We're going to do neurological exams and things like that, but we might not have a handle on the genotype at that point. That's one of the problems with DNA sequencing is we don't know what all the variants mean. I think you understand that. One of the advantages of Amy's approach is she's looking at yeast growth as a readout. So here, if the enzyme is non-functional for any reason, like it's not folding or it's folding but the active site is broken, she will pick those up, okay? So it's better to have a functional assay rather than a folding assay. And so we always look for ways to read out these cells with functional assays. Let's say you have a fluorescent substrate that goes into cells and becomes fluorescent if the enzyme is active. We can do the same thing. We can sort cells based on fluorescence, now based on activity. But for the ARSA, we didn't have a fluorescent assay. All we had was the abundance of the protein through the fluorescent protein tag. So we measure abundance, which is usually good enough, but not always. Amy measures yeast growth, which depends on the enzyme being functional, okay? It's better. It might be a little lower resolution. I don't know, whatever, there's different ways we can make libraries of cells that have one variant per cell, and then we have to score them somehow. Okay, I'm not gonna talk about the area. I wanna finish in 5 or 6 minutes. In your lifetime, right, in the next 5 or 10 years, we're gonna be, if not sooner, unleashing whole genome sequencing of newborns. Right now it's being done at pilot studies around the world. Potentially very powerful because we can track, let's say, 600 early onset childhood serious and treatable diseases before it's too late. We certainly can't do that many with biochemistry. On the other hand, the challenges are enormous, and they're so enormous and so unpredictable that I don't know whether this is going to implode or not. And anybody who says otherwise, I just don't believe them. Here's one of the problems that makes me very nervous, but maybe surmountable. You know, we call variants pathogenic in ClinVar, according to American College of Medical Genetics guidelines. It means it's been seen in a patient, there's case reports. It is a driver of the disease. It's pathogenic. But take a disease that's recessive. Suppose you have there's different flavors of pathogenicity. Let's say you have a variant that totally wipes out the protein, and it's paired with a missense variant that partially wipes out the protein. You might have a late onset disease that's driven lateness by the penetrance or the residual function of the partially disrupting mutation. But if you have either one of these mutations, you don't get the disease, you're a carrier, even if you only have the severe mutation, because 50% enzyme is enough to keep you healthy. But they're both equally pathogenic without distinction, because because they both have been seen in patients and you need both of them. So they are drivers of the disease, not in the test tube, but in humans. Now, what happens if you have a late onset with another late onset pathogenic? And the answer is indeterminate often. You might not even get a disease because 5% plus 5% is 10% function, and that's plenty to keep you healthy. So the sequencing people are going to call out all cases of pathogenic for recessive diseases. Okay, fine. They're not going to call out variants of unknown significance because they don't know what to do with them, and most of the time they're benign. But among the pathogenic, most of them are going to be false positives. Okay? Let me give you a real story. So we've looked at 470,000 human sequences in the UK, United Kingdom Biobank. We've already sequenced 470,000 humans. We have their sequence, whole genome per human, not just a bottle of alleles, but the human genomes. MPS one example, a disease, a lysosomal. We find 30 patients in 470,000 that have two pathogenic variants. They're likely in trans, even though we don't know the phase, because that's the way it usually works. They're rare variants. And none of these people have MPS one. Okay, so pathogenic does not equal disease. So we have a big false positive problem. Now, what about the false negative problem? We're not going to be calling out VUs, so we're going to be missing cases. They would argue, well, it's better to catch half the babies with these 600 diseases, even if we miss half, because we're screening for 600 diseases and we could save 300. I agree with that. In biochemical newborn screening, the way we do it now, a single false negative in the state of Washington per year is considered taboo. But if we do whole genome sequencing, the false negative problem will be massive and we accept it. Although we'll see if the parents complain about false negatives when their neighbor down the street was treated, but they weren't because of a false negative. So can you answer whether this will be acceptable to society? I cannot. So the false positive problem, no matter how you look at it, is massive. The false negative problem, probably less massive, but concerning. So this technology is coming, and what's going to happen, I would say nobody's smart enough to know. It could be partially successful, or it can implode, but it's not going to be smooth sailing. It'll get better with time. For example, once we sequence 20 million babies and learn all the healthy babies that have two pathogenic variants, we might be able to make a list to say, if you have that genotype, you're okay. We might be able to do this by so-called federated learning, by taking all the data we have and eliminating, filtering out all the concerning diplotypes that we know are not disease, even though you have two pathogenic variants. So maybe that'll take 10 years. I don't know. Nobody knows, and we'll be okay. I'm going to end with one more technology that I'm very excited about. I'm going to talk about a disease called cystinosis. It's a rare lysosomal storage disease. You're missing a transporter in lysosomes that pumps cystine, you know, the dimer of two cysteines, out of the lysosome when it's formed by protein breakdown. That's the only thing this transporter does, is transport cystine. And if you're missing this, you have cystinosis. We can treat it pretty well. It's a transporter. We can't measure its function in a dried blood spot. No way. I need to. The way we treat it is remarkable. This is cystine. We treat it with this drug called cysteamine, very simple drug. It opens up the disulfide to make cysteine, for which we have a different transporter that works. And this thioether analog of lysine that the lysine transporter works on. So we can clear cystine by adding cysteamine. It's a very clever treatment that was developed at UC San Diego. So the game plan here for biochemical diagnosis or newborn screening of cystinosis is to measure the abundance of the transporter. Most of the time we've done this, we think 90–95% of the cystinosis patients have a deficient amount of this transporter because of a missense mutation or a deletion or a stop codon. But after 30 years of work, nobody has been able to do this by ELISA because they don't have a very good antibody. You might be able to fix this, but anyways, it might work. We can measure prostate-specific antigen, with ELISA is just fine, low abundance. But let's come up with a way that I think is going to work better, and I think could sort of revolutionize the way we do clinical proteomics. The way we do proteomics, right, is we take the blood and we treat with trypsin to generate a huge number of tryptic peptides, right? Each protein is giving 20 to 50, 100 tryptic peptides, and we can detect these by liquid chromatography tandem mass spec. This is how proteomics is done. The problem is, for low abundant proteins, it's a needle in the haystack. You're trying to find the cystinosin-derived peptide in a massive number of peptides with higher abundance. You'll never find it. You need some sort of enrichment. This would typically be done by taking an antibody to the peptide and pulling it down on beads and then detecting it by mass spec. But now we can design antibodies, essentially. With David Baker, who just won the Nobel Prize, the current Nobel Prize in Chemistry for 2025. Kejia Wu is a postdoc. We can design proteins now that bind your favorite peptide without exception. We've done this in the science paper that just came out, we've done this on about, 50 proteins, and we got high affinity design peptide binders in 48 cases on the first try. So essentially, it always works. Now let's think about designing a protein from scratch that binds your favorite peptide of any amino acid sequence. This is a pretty complicated jigsaw puzzle in 3 dimensions, right? We've got to make, I don't know, 15 hydrogen bonds from the protein to the backbone of the peptide and polar side chains of the peptide. Then we have to make all the hydrophobic stuff fit as well. And this can be done by machine learning based design without any physics. It's just we train large data sets of binders, and genetic algorithms can learn the parameters, the dial settings that are needed to generate from scratch a de novo peptide binder. This can be done in a matter of hours, and then you would make, let's say, your 30 top designs in E. coli. That's trivial now. You order the genes, you put them into E. coli, and then with high throughput peptide binding assays based on optical techniques, we can, out of 30, we essentially always get 5, 6 proteins that bind the peptide with nanomolar or subnanomolar affinity, which is what we need for proteomics. This works. Let's see, I'm going to stop in a minute or two. Anyways, we can find the cystinosin-derived tryptic peptide after we capture it with our design protein and do LC mass spec. We see this in normal people, but when we looked at 10 cystinosin blood spots, we did not see this peptide. So how do we know this peak in the mass spec is our peptide? Remember, there's billions of, maybe trillions of peptides. Well, it has the right chromatographic retention time of our peptide. We can synthesize the peptide and show the authentic retention time. It has the right mass and it has the right mass fragments. Okay, but perhaps the strongest evidence is in human knockouts that don't have the protein. We don't see the peptide, right? That's the best evidence. It is not a ghost peak. It is cystinosin derived. Yeah, so this is Amy's stuff. Now, imagine you could do this on hundreds of diseases. For example, we can do this on Wilson disease. Wilson disease is a copper transporter in the Golgi, super treatable with a simple drug, and 1 in 20,000 babies. Can't measure the function of a copper transporter, but we can do it by targeted proteomics. Think about, let's say we start sequencing babies, and we have lots of maybes for lots of diseases. For most of those diseases, we think knowing the abundance of the protein in a blood sample or a muscle biopsy or whatever we can get, we think the abundance will be way down. So one of the first things you might want to know if you're worried about DNA sequence result is the abundance of the protein, okay? So we think this could be a good add-on to DNA sequencing first here. And it can also be used as a first-tier newborn screening biochemical, like I showed you for cystinosis and Wilson disease. So I think this is going to be powerful. We would typically do this with monoclonal antibodies. Sometimes you get a good one, sometimes you don't. It can take a long time. It gets very expensive. We think we can design proteins at will now that bind whatever peptide you want from whatever protein you want. And it's just a question of whether you can get a biological sample that would have the protein in it. Is the protein expressed in blood cells, etc. But we think we can do a proteomic assay on just about anything now by having designed peptide binders. Okay, I think I'm going to quit there. We're at the 1 hour mark and open it up. So maybe this is coming in a big way for diagnosis and newborn screening. This is brand new. He's starting to talk about it. Paper just came out. Let's see what happens. Okay, Amy, I think I'm going to stop there and see if there's questions for Dean or I.
Thanks, Michael. And thanks, Dean. That was great. So people should feel free to unmute and ask a question, and it'll be easier if you also come on camera, or you're welcome to post questions in the chat, and I'll monitor that. Maybe I'll take the liberty of asking a couple of questions to get us started. One of the, Dean, you were talking about, actually you were both talking about kind of the gene therapy that's available now for MLD. In some of the metabolic diseases that I'm most familiar with, there's still there's still research space for kind of what is going to be the gene therapy winner, right? Is there going to be a place for mRNA-based gene therapies? Are we going, are we talking about enzyme replacement? Are we talking about prime editing? Do you see, for example, and sometimes this is an issue around kind of, what do we do with the neonatal onsets? In the case of OTC deficiency, for example, there's neonatal and then there's later onset, and might you want to have multiple therapeutics simultaneously? Is that something that, just getting a gene therapy approved, that's great and that's awesome, but do you see a space for multiple types of these kinds of therapeutics in your disease?
Yes, and I chuckle because you say just getting a gene therapy approved.
That little thing.
I was so much more hairy back then. No, yeah, there definitely is room for a number of reasons. I mentioned and this is not unique to MLD. I'm sure other disorders have the same issue. This therapy is it's very effective, but it's also complicated. And so there are only 5 treatment centers. We're working, the greater community is working on a 6th treatment center in the US, but that means that, the people in New York don't have a treatment center. They're going down to Philadelphia, or they're going, elsewhere and so on. So even in places you think might be focal points, they can't all be. So there's opportunities to fill in those geographic areas, and certainly different forms of infusion or therapeutic application. We completed a long-term enzyme replacement study, and families are still on that. I think we're 3 years post therapy and post sponsoring company decision to say this clinical trial was a success, and the answer is no, this doesn't work. But the family thinks it works, and it was a therapy, at least in my opinion, is a clinical trial design problem, saying, the endpoint wasn't, maybe that wasn't the right endpoint, So and those of you that do clinical trials know you set those up years and years in advance of seeing the patients and seeing all the end results. The other thing that is an opportunity is, while this is built off of an autologous stem cell transplant, people are looking at other types of infusions, be that different injection sites or different injection mediums. And we're also working with a company that is based in Brazil that has an innovative business model that allows them to be, we hope, to be much more successful. Hopefully, they're successful on the science side, but also on the business side, meaning they can serve the other 90% of the world that doesn't live in the US and the EU, right? And there are a lot more cost considerations when you go to those broad populations. The gene therapy we have right now requires that the modified cells be frozen and returned back. Well, they're frozen both directions. They're still being manufactured. And we smile when you say we're manufacturing genetics, but in Italy. And you can't do that. And I'm being very broad brush, but you can't send that to the bush country of Africa or the jungles of South America. So there's plenty of opportunity for improvement. MLD is the world's most expensive. This gene therapy is the world's most expensive therapy today. It's $4.25 million list price. Mind you, before you choke too much, that's seldom what anybody pays. Medicaid, which is where most of these patients are going, gets an automatic 20%-ish discount, and down it goes negotiated below that. But nevertheless, millions of dollars is a lot of money. So we have to be thinking about the economics, not just of MLD. This is dozens of patients a year. But what happens when, Michael is screening for 600 disorders and, we get hundreds or thousands of patients per year. Our health system is not set for that. So that's a long answer to say there is plenty of opportunity to be creative and take on different approaches.
But, okay, the cost is complicated. For example, the United Kingdom is on a national healthcare system, right? Government, single
Right.
payer. And their committee of experts approved the gene therapy for MLD, probably after long discussions. People have done analysis on the cost effectiveness because, when kids have MLD and they're not treated, it's also expensive, millions of dollars to care for them while they die. So there's two kinds of, is a treatment cost savings. It means that it costs the healthcare system less to treat than not to treat. That's not the case for MLD. Is it cost effective? That means the cost of the treatment and the follow-up is considered against the cost of not treating, but palliative care and follow-up, and how long do patients live. So the healthcare systems have a certain threshold, right? That it might cost more. It costs net dollars to treat than not to treat, but it's below the threshold of what insurance companies and healthcare systems are willing to pay, right? So if you get $1 million drug for cancer that keeps you alive for another month, it's not going to pass that litmus test. But for MLD, it well passes that test. Okay.
Agreed.
So
Agreed.
Now the question is, if we have gene therapy for 500 diseases over the next decade, are the thresholds going to go down because we can't afford 500 gene therapies? But assuming we don't just ignore these babies while they die, right, which I think we'll never do, or we might, it's going to be better to treat according, but we could run out of money. I don't know what to say. We have certain current day thresholds on cost in versus not treating, and it passes that test. But what happens if we start treating lots and lots of rare diseases by gene therapy? Those thresholds might change, and nobody knows. But right now they pass. Okay, that's the best way to put it.
Oh yeah, and I didn't mean to say
Yeah,
what we have is not cost effective, to put this in an ROI, return on investment sense. But you know what? If the therapy was half the price it is today, it would
Right.
free up money to do
But
something else.
you could. I have people at the end of talks say, you can do a lot more with $4 million than treat one MLD baby. I'm not getting into that, guys, because
Different
we don't ignore babies that are not treated. We don't say bye-bye and ignore them. We pay money. So anyways, this is a big question, and healthcare systems have thought about this, and they've made decisions based on these current thresholds, and that's where we are now. So I have no answer to the guy that wants to spend the $4 million on something else to help, unfortunate people out there. I have no answer other than it's a big question that's been thought about by healthcare systems, and they've made decisions.
I just wanted to raise a question that was posted in the chat by Walter Russo. Do either one of you care to speculate as to whether disbanding of the HHS committee upstream of the Rust reflects hostility to newborn screening or general hostility to science, or perhaps just short-sighted frugality? Do we have any?
We don't really know, except it was done during the Doge, whatever Elon.
It was at the very beginning, right?
So it seemed like an obvious target because it won't cost a lot of votes, I think. I would say.
No, well, and
But
I'll just
yeah, that's my take. Is it attack on science? I don't know. I guess so, maybe. But it happened. All I can say is it happened during the government, machete phase.
Yeah, it was very early.
So I have a slightly different perspective. I mean, all that is true, but the committee itself was a provisional committee, and I think 7 or 8 other committees that were provisional, meaning that they didn't have formal authorization and reoccurring budgets. They were discretionary. I say provisional, discretionary committee that the prior Secretary of HHS had kept going, and Kennedy was as well. And all of the provisional committees were swept out. I don't believe that newborn screening was targeted. Honestly, I don't know Secretary Kennedy, and I haven't heard this from anybody, but I suspect that in the scheme of things with the NIH and Medicare, Medicaid, CMS, all those FDA, all those kind of big ticket items, newborn screening is not even a drop in the bucket. It's a mist in the air of expenses to him. So I suspect he just has or the staffers that allow this to happen had no idea. So I don't perceive it that way. I can tell you that as we're working to get DMD and MLD separately, but in parallel, up to Kennedy now, his staff is very responsive and has been very positive and acknowledges the impact of newborn screening and the curative nature of our gene therapy, and is very receptive. We just closed, in fact, they established the Federal Register comment period or a comment period on a Federal Register posting that just closed, I think this past weekend or maybe on Monday. And that was what we think is the final decision point or data point that he will use for his decision. But so I don't feel targeted, but nevertheless, in the bigger context of how funding's being looked at and how rules are changing and those sorts of things, it's not inconsistent either.
Kind of along the lines of funding and healthcare systems, Sylvia Lucas has a comment and a question in the chat. How do countries with universal healthcare over the population cover these children, i.e. in the EU countries?
I mean, I don't know, but all I can do is tell you the UK has approved the gene therapy for MLD, as has Germany and Austria and Norway. Holland, I think, has not yet. So it's not universally covered across Europe. How is England going to pay for this? I don't know. I would say England is probably at the forefront of whole genome sequencing of newborns to catch rare diseases. Why is that? I don't know. They have Genomics England, which the federal government and the healthcare system is pushing for this. They think it's going to do some good for kids and for the economy. I don't know whether that's true. I don't know how much Illumina sequencing is putting money in. I went to the newborn sequencing meeting in London, and one of the head government officials who suffers from a rare disease thought it was a good idea. I don't know the answer to Sylvia's question. Nobody does. Maybe somebody does. I don't know. I don't know.
So Sylvia, I would caution, and I don't know the answer either, but when we say just let the government pay for it, that means all of us are paying for it. So whether it is the healthcare system or private payers or public payers, at some point we are all paying for it because that's how healthcare is paid for. If it's private insurance, that means all the people on the private policies that are healthy are paying for those that aren't, just betting that they'll have insurance when and if they get sick. So it is, and this is one of the things that's changed in newborn screening. Newborn screening used to run somewhat independent of the payers. And officially they do. It's just like the FDA is not supposed to be involved in the cost of therapies when they go through the approval, but they ask questions and they touch up against that. And when you have a public health system that's identifying a patient that, in the case of these infantile onset disorders, of which most of the newborn screening conditions are, they will end up on Medicaid or already on Medicaid, in our case, over 50% of the babies. That means that they're identifying a patient they're going to have to pay for. And that creates a little bit of a conflict of interest. And so what we've seen over the past, and I think there's pretty good arm's length, but I've seen those conversations happen. I think that highlights what one of the transition points in newborn screening is a lot of these ethical discussions. The ethicists are having a much bigger voice in how we think about these things and what is fair and what's reasonable and what are the, you can't just be an accountant and figure out and decide who gets to have a therapy. Whether you have a positive score or a negative score, it can't be just an accounting thing. There's got to be a variety of other criteria. We were part of the ICER and the NICE reviews. They turn everything into ultimately into dollars, quality adjusted life years. Someone that is sick is not worth as much money per year as somebody who is not. And that really, really doesn't work for me. It happened to work for MLD, but I think in the big scheme of things, we have to be more have more perspective than just how do you relate everything down to dollars, because we see. I took a very quick vacation down to Cabo San Lucas. Had never been down there, far southern tip of Baja Peninsula, celebrating my daughter's 45th birthday, the MLD daughter. And we met a lady while I was pushing Lindy up and down some streets that weren't meant for wheelchairs. A lady pulled over, a young lady, and asked me, says, where did do you have a wheelchair van? Where did you get that? And this and that. And she had met a gentleman about 3, 4, 5 months ago, and he is a quadriplegic. And she was attracted to him, God bless her. And I mean, I imagine there's a care burden. I know there's a care burden for that. But his quality of life, certainly he's not running marathons and he's not climbing stairs, but he has a life and he is cognitively alert. And yet there's a dollar penalty for the fact that you can't walk and you can't run. And so these things are I'm sorry, I'm really going sideways here, but these things are very, very complicated. And the answers, unfortunately, aren't as black white as we'd like them to be. In fact, Mike started off with saying it's gray, and I completely, and he's talking about sequencing and scales and measuring and all that kind of stuff. It's gray, and I think that's the world that we live in.
I also want to say, I'm not a health economist. I don't know these numbers, but there's more to it. Sylvia says 2.3 million for each SMA's patient. Sounds expensive. Sorry, it's not that simple. Sounds expensive. I don't know what Sylvia thinks. Take the number of people, take the cost we spend on Enbrel. I don't know, what is that, $20,000 a year? Divided by the number of people that have rheumatoid arthritis to take Enbrel in the United States. And compare that to the cost for SMA divided by the number of people that have SMA in the United States. Let's do that calculation. Let's not forget how much money we spend on Enbrel. On the other hand, a lot of people take Enbrel, and we're saving a lot of people. So these you can't just say, it's like in newborn screening, you got to buy a mass spec that costs $500,000. That's a silly thing to say. The answer is, how much does it cost per baby? And the answer is 50 cents. Now you have the tough question, is 50 cents per baby worth it? And I don't know the answer. But it's not $1 million, right? You can't say $2.3 million. It has to be normalized to the number of people. In Australia, I think they have a rule that no single person deserves more than a certain maximum dollar investment from healthcare. In the United States, we don't have that rule. We have this threshold. Which system's better? I don't know. You might say it's more futuristic and more fair that everybody is held to a certain number of dollars per person and can never go over that. Nobody deserves more dollars than anybody else. I don't know.
Yeah, Sylvia, we're not challenging you or
Yeah, I
meaning
know.
to.
Nobody knows the
It's
answer
a
to this
not. It's
question.
it's just not a
Oh,
clear
no,
answer.
no. This is
Yeah.
Sylvia. Can you hear me?
Yeah.
Yeah,
Yes,
I don't know what Sylvia's thinking. Sorry.
I'll tell you what I'm thinking.
she's gonna tell us. Yeah.
I mean, the science is fantastic, and thank you both for wonderful talks. Um, in 5 to 10 years, we'll have whole genomes of all the newborns. That's great. I happen to be on the pharmacy and therapeutics committee of a very major insurance company, covers millions of lives, and so when the first gene therapy comes out and the scientific company that's selling it offers to put the insurance company on a payment plan, that kind of, opens your eyes a little bit, because if they start not covering people at high risk, which will probably happen, or if I mean, you're absolutely right that all of us will bear the cost of this, but at the risk of insurance companies, for example, increasing the price so that people can't afford insurance, being dropped, that kind of thing, and especially now. And I don't want to get into the politics because my blood pressure always goes up, but it would be great if we could have, and maybe you're right, we should all write letters, but it would be great to take the cost of not just the screening, not just the drug, but also the care, the lifetime care of these people, and spread it out in the federal government, or whatever passes for the federal government these days. Maybe we'll have to wait, but I don't know how else to approach that, because I'm worried that other people will not have coverage, and
Yeah.
because
What do you mean? What do you mean? Pass it through the federal government, in a logical way, with assuming that's possible. But
A risk pool
what do
or
you mean
something.
by passing it through the federal government?
Well, let's say metachromatic leukodystrophy.
Yeah.
You if let's say we have people that live through that, which we are going to have that with gene therapy, it's still going to be expensive. So would Primera or Regents or Optum pay for that, or should we have these kids automatically enter a program like Medicaid you brought up? But is Medicaid still going to be around in a while? But I just think
Oh, yeah.
I just think we need to pass those costs on if we value people. Now you're right about qualies, because sometimes it's an affront to think these people are only worth what they can make in their job or life or whatever. But and we're facing the same thing with Alzheimer's patients. You get diagnosed at 70 or 80. Well, what's your quality then when you're retired and things
Right.
like that? So I agree. That's difficult. I don't know how else to do it.
Yeah, I think this whole health economics and qualities and how much a human life is worth based on how much you can make if you work, or how much you're willing to spend on life insurance or critical, auto insurance. Your life is worth how much you're willing to insure it for. That's how they do it, and nobody finds these arguments very. I don't know. I'm not an expert. Maybe they're reasonable. They sound pretty bad to me. So we don't know, but maybe you're an insurance company. How much is a human life worth? I don't know.
Yeah, so two other quick comments just to add to that, again, just generally for the rest of the audience. So particularly in private insurance, there is reinsurance. So many of us have heard of Lloyd's of London. They insure the big ships or whatever. I don't know what they do.
Right.
But if the expenditure is above some number for your local insurance company, even if private, even if it looks like a brand name, they have reinsurance where they're spreading that out into a broader pool.
That we all pay for it.
We all pay for it, but it's spread out, pennies to all of us instead of tens or hundreds or thousands of dollars to a smaller pool. The other thing that is happening, and again, this is just the whole economics is.
Yeah, but then you add up all the pennies, right? Because
I get it. It's still the same amount of dollars, but it's
there's not a lot of MLD patients. So if you add them all up, it starts to become more than pennies. But it's complicated.
the other thing I want to just mention, because we haven't thrown under the bus, so to speak, the pharma companies, because they do set these prices, and they are the ones that are spending and funding a lot of these programs and investigations and so on, bringing products to market. They are actually putting proposals forth where they will guarantee, they're not using the word guarantee, but it's a contractual guarantee that says if you pay $4 million for an MLD therapy, again, they don't actually pay that, but it's but millions of dollars. If it doesn't work, if that child does not meet the outcome criteria that we agree on, we the payer and the provider, that they will refund back some of those dollars to the payer. It's like, so having them in the boat with the families, and guaranteeing the products, kind of like an extended warranty on your car, I guess. And that's a crappy example too. But you know, there are a lot of creative things that are going on. None of them are, they're not intuitive. They just, it's like we have to figure out who we're going to be as a society as we continue to grow up. And as these things change and we look at, you know, it's just amazing what we can do with medicine these days, and yet it's not free. So we need to figure
one thing that really opened my eyes at the World Conference on Lysosomal last year, and there was a guy saying that we have this therapy for DMD. I forgot which therapy. It's been around for a little bit, and it costs millions of dollars. And Duchenne's is pretty common. And it's very incremental therapy. It's just not worth it. And the company, they never rescind on the drug. And they're calling for them to rescind, but there's no law that says you have to. I don't know how it works. But the numbers were so grotesque that we should pull this drug off the market, but it never happens.
Yeah.
And MLD is nearly curative for a disease that is the most horrific thing you've ever seen, where a kid loses the ability to walk, talk, see, think, hear in the first years of life before they die. And now they're going to high school. Okay, that's not incremental therapy for Duchenne's muscular dystrophy. That also costs $1 million. So yeah. I think companies should rescind when the drug is just barely incremental, because a little bit better is not always better, right? When you consider the costs. And
so I'd like to end on that positive note about how
Thank you
curative
for answering
MLD
those
is.
questions.
Yeah. And I'd like to thank everyone for joining us for Science Matters. And I'd especially like to thank both our speakers for taking the time out today and for providing great talks and a stimulating conversation.
Thank you for having us. Really appreciate it.
Thanks,
New
guys.
Screening Walks.
Thanks, Amy.
See you later. Bye.
PNRI.
See you both later.